Okay, well, good afternoon. This is a picture of St. Patrick, who's the patron saint of Ireland. The rumour has it that he banished the snakes from Ireland in the 15th century, and here's a picture of a rather angry-looking St. Patrick telling them where to go. Now, whether that was the reason, or as some believe it was the Ice Age that killed all the reptiles out on Ireland, not to be repopulated on the island, whatever the reason, Ireland does not have snakes. My wife is Irish and my mother was Irish, and both were terrified of snakes their whole life, despite the fact that snakes did not play any part in their life in Ireland. And I guess you can imagine how it may be an evolutionary advantage, given that we've shared the planet with these venomous creatures for 90 million years, that our primate and shrew ancestors might not like snakes. But with good cause. I won't go through the numbers you've heard today. 100,000 people a year dying from snakes, 400,000 sequelae, and so on. But just to before I get into talking about some of the pioneers, apart from St. Patrick or the Ice Age, a number of people that can be credited with making a big contribution in this field. It all began back in the 1890s in France, and in fact there were two groups at the same time generating anti-venom antibodies. Cesar Ficelix eventually went back to other interests and died young, so most of the credit for this goes to Albert Calmetti, who was famous for creating a tuberculosis vaccine, but also was the first person to generate equine antibodies against the Indian cobra. The baton then was picked up by Vital Brazil, who you heard about in the last talk, who again picked up from Calmetti and continued to develop a number of anti-venom, polyclonal anti-venom sera. I'd also like to mention Clodomiro Picado, who was coming behind them. Again, in Costa Rica, there was a big toll of death from snake venom. He, again, imported the stuff from Brazil and set up to make these venoms here. So both from Brazil and from Picado, both formed institutes, both still making antivenoms today. But of particular interest is the Institute Clodomiro Picado in Costa Rica, who are my important collaborators in the work that I'm about to start at the University of Cambridge. Again, different people have addressed the problems of polyclonal sera in different ways. We heard Nick talk about the variation in the composition of the venoms based both on region and actually also on the age of the snake. We're also dependent on the immune response of a horse or whatever animal has been immunized. And of course, again, we heard in the last talk how that can be variable in itself. Once we've generated the antiserum, only a fraction of the material is actually relevant to some of it. The antibodies will be to non-venom targets, and some of them will be to components that are not related to the toxicity. Some of the immunogens or some of the antigens will be of low immunogenicity. So to neutralize those, you just have to pile more and more antibody in. And so the poor unfortunate who's just been bitten by a snake may end up having to suffer 15 grams of foreign protein being injected. So if the venom doesn't get them, the anaphylactic shock might get them. So really, a few years ago, I became interested. And so I was one of the founders of a company called IONTAS 10 years ago, which I sold 2 years ago. And during that time, there's two sort of aspects of venom therapeutics that became of interest. One is the idea that venoms themselves might actually provide drug molecules or drug leads. And indeed, there are many venom components that are used to block ion channels. And so that was an area of interest, and we developed the technology wherein we would fuse small cysteine-rich peptides into the surface of an antibody to create antibody-like molecules that blocked ion channels in a way that was also amenable to phage display and mammalian display selections. So I won't say any more about that, but Anish will talk about this in tomorrow's session. But the other area in the subject of today is the idea that we can make recombinant antibody venoms that are more prescribed oligoclonal cocktails. And a couple of years ago, I applied for and got a Wellcome Trust grant, which I'm now just activating at the University of Cambridge, just in the very early stages of setting that group up. And again, as I've mentioned, that I'm working with Andres and Kima at the Institute Claudemiro Picado to take the horses that they've immunized and isolate antibodies from those, but more of that later. But I'd like to begin by just giving a few sort of stories around work that we've done with human antibodies and human antibody repertoires to sort of set the scene. Again, you saw this slide earlier on today, and it's again from Professor Gutierrez showing the relative frequency of different components in venoms of elapids and vipers. And suffice to say that there's a tendency with the elapids for these neurotoxic venoms made up of dendrotoxin and alpha-neurotoxins, and with the vipers, often enzymatic products causing necrosis and hemostatic effects. So in the next few examples, I'll talk about the examples within the three ringed areas here. And the first of these is the metalloproteases. So there's the P1 metalloprotease, which are typically just an isolated catalytic domain. The P3 metalloproteases, such as this Russell Viper example for which the structure has been determined, have the catalytic domain shown in pink, a disintegrin domain and a cysteine-rich domain, which maybe contribute to substrate specificity. And in this particular example, it cleaves factor 10 to give factor 10A, which in turn activates prothrombin to thrombin, causing clotting. So we haven't actually worked directly on the Russell Viper metalloprotease, but we have worked on mammalian proteases, which are phylogenetically linked. So you can see next to the Russell Viper structure, the structure of ADAM22, which is a membrane-tethered protease. In fact, many of these ADAM proteases in mammalian cells are referred to as shedases. They sit on the cell surface and are involved in the liberation of growth factors and cytokines. And we're particularly interested in ADAM17, also known as TNF-alpha converting enzyme, because of its role in activating TNF, also known as TACE. Again, a structure for the catalytic domain has been determined, but not for the full ectodomain shown on the far right here. So the goal in this project was to actually make a blocker of the ADAM17 TACE metalloprotease. I should say in the decades before this, there'd been a lot of effort to try and make blockers of proteases for therapy with humans, but all of them, or most of them, failed in clinical trial because they weren't specific enough for one individual protease over another, which I guess from the point of view of targeting venom proteases is probably a good thing if we can create such cross-reactivity. But again, in this particular example, the goal was to make something that was as specific as we could get. So we carried out phage display selections on the whole ectodomain. We incubated with a protease inhibitor and went through several rounds of selection and identified one in particular that we focused on. It's this D1 antibody shown here. And on the left, you can see a proteolytic assay with a tethered fluorogenic peptide. And we can see that straight out of our naive library, and this I should say is work that was done at the University of Cambridge when I was in the biochemistry department. We can see that this antibody is as good as the TIMP3 control which we were using. When we actually look at the specificity of this antibody, if you look on the graph on the right, you can see in the middle a polyclonal antibodies binding to both the whole ectodomain as well as the catalytic domain, and those two lines are overlapping. When we take the individual antibody, we can see it doesn't bind the catalytic domain at all, so it's binding something on the ectodomain. And through various studies that I won't go into, we determined that it was largely heavy chain driven to the dis-cis domain. So we then set about a chain shuffling example, so introducing diversity, keeping the heavy chain, introducing diversity with the light chain, and looking for an improved antibody through this standard chain shuffling method that we use in the phage display world. And so we were able to do rounds of selection and generate an antibody which improved the affinity from the initial 26 nanomolar of the parent to 0.4 nanomolar. And in doing so, we found a new light chain partner that enhanced the binding of that heavy chain. And in addition, the antibody now bound to the isolated catalytic domain. And again, through various mutagenic studies, the model is one wherein the heavy chain binds to the dis-cis domain and the light chain binds at the catalytic domain, making this very potent neutralizing antibody. So, of course, that's not a venom protease. I give it as an example of being able to use phage display to make blockers to proteases. And in fact, we had quite a successful collaboration with Jill Murphy's lab at the university, leading to another 4 publications describing the generation of protease blockers. Okay, I'd like to move to a new example, this time tackling dendrotoxin of the black mamba, a dangerous snake with a very fast-acting venom consisting largely of alpha-neurotoxin and dendrotoxin. So Andreas came to the lab about 6 years ago and worked closely with Anish shown here. And he did actually used to look that young before he started working with Andreas. And this is also grateful for Andreas for bringing this project and idea to our lab. So the idea was that we would fractionate the venom of the black mamba. We would biotinylate the antigen, use it to drive specific phage display selections to those components, and then characterize the antibody. First of all, screening for binding and also using an affinity capture assay to really work out the highest affinity antibodies, converting to IgG format, and then doing things like mass spectrometry to confirm the binding to the antigen of choice. In this case, most of the antibodies coming out of this were to a delta dendrotoxin molecule. And so the next step was to take that in vivo. In this case, we used a slightly unusual intracerebroventricular injection method to show the blocking effect of dendrotoxin on the voltage-gated potassium channels, which kind of act synergistically with the neurotoxins. And this was work that was done both by Erska and Cecile, at least the preparation of that material. And the end result was we were able indeed to show protection of these mice, either with individual antibodies or even preferably with combinations of those antibodies. And we published in this paper back about four years ago. Third example, again at the time Andreas was there, he initiated some work on the alpha-neurotoxin of the monocled cobra. And this work was then picked up by Leni, shown here. So this is again long-chain alpha-neurotoxins, which make up about 35% of the venom of this species. This molecule is an antagonist of postsynaptic acetylcholine receptors, and it again just blocks the channel, causing neuromuscular paralysis. And again, Leni picked up from where Andreas left off, and we've just had this paper accepted in MAB, so that should be with you soon. But returning to the initial work that Andreas did, we had this panel of antibodies that we converted to IgG. You can see on the left-hand side that the control mice were dead within about half an hour, whereas the antibodies that he created extended the lifetime. It didn't prevent lethality, but it extended the survival out to about 6 hours in this case. So Leni came in and again took this chain shuffling approach, fixing the heavy chain, bringing in a repertoire of light chain partners and driving stringent selections to identify a new panel of binders. And shown on the left in one color is the direct binding by single chain Fe, shown in gray, but also shown in blue is again this affinity capture assay we use, which is much more discerning for affinity and helps to really identify the best guys. So these single chains then were converted to IgG, and again we can see another affinity capture experiment shown on the right-hand side. And then incidentally in both plots we can see on the far right the original antibody that Andreas had generated, so you can see a significant improvement in the binding of these. This led us to this antibody with the long name shown there. We were able to measure by Biocor and show an 8-fold improvement in affinity to somewhere around 0.5 nanomolar. We also set up a receptor blocking assay using immobilized nicotinic acetylcholine receptor binding to alpha cobra toxin, labeled alpha cobra toxin, and then pre-incubating with antibody to demonstrate blockade, which again we can see for both of the antibodies with an improvement on the affinity improved version. And then finally, working with Sophia and Lene and Andreas, did a direct neutralization assay measuring by electrophysiology the consequence. So first off, there's a titration of acetylcholine to identify an EC80 value. Using that AC80 value of 70 micromolar, they then did an alpha-neurotoxin titration to look for an IC80, and then measured and demonstrated the blockade with the antibodies, as shown on the plot on the right-hand side. So again, this is now taking those antibodies in to the in vivo assays now by intravenous injection. And again, we can see with either the venom or a nivolumab control that the animals are dead within about half an hour. And shown in green is a polyclonal antisera that we had. So for our antibodies, we then incubated them with the cobra toxin at different ratios before introducing them. And now we can see that the plot previously on the left is now shrunk into this area shown in the circle, where everything was dead within half an hour. And now we can see that with increasing doses, we've increased the survival of these mice out to the 48-hour time point, and in fact, in the highest concentration ratio, those animals sort of survived past the end of the experiment. Interestingly, the parental antibody, if I remind you, was the death at 6 hours. So through this combination of through affinity improvement of this antibody, we've been able to convert from a kind of extended lifestyle to an inhibition of lethality. And for me, the big take-home message of this, need it be said, but say it anyway, affinity is very important to this. And especially when one's thinking about trying to make an oligoclonal cocktail where any individual antibody may be given at lower concentrations, being able to drive the affinity to really mop things up with this whatever limited concentration you have within a cocktail, I think is hugely important. And so that for me is what made me, despite having worked in these early stages in fact for the last 30 years with naive human antibodies, it kind of occurred to me that really there is mileage in trying to generate antibodies from the immune repertoires here to try and capture that affinity benefit. So I'm collaborating with Institute Clodomiro Picado, who actually make a polyclonal antibody for sub-Saharan Africa to the 3 snakes shown here by immunizing horses, caprylic acid precipitation to remove non-antibody protein, et cetera, et cetera. And so the idea is that we can hopefully tap into the benefit of this. And again, this is a very detailed slide, but it's simply to say that in addition to the purification step, which is very carefully controlled, they also do a lot of validation of those antibodies. And we can see here different measures using mice to look at lethality, hemorrhagic effects, coagulation, and necrosis altogether. And in this particular example, the study was looking not just at the two snakes that were used as a source of immunization, but also related species. And again, this table is showing that there were indeed good neutralization to other snakes beyond the one that they were immunized with. So the whole sort of thinking here is that we'll immunize horses. Of course, we all know with horses there's a heavy light chain combination problem, but in addition, llamas to where there's the affinity is driven by a single chain. We'll clone the repertoire, we'll make phage display libraries from those using sort of standard techniques shown here, how we make our libraries, in this case for the horse. So again, the idea is then we'll take these, so I'm going through this rather quickly. The idea here is that we will take the libraries we've created and we'll either select on the purified protein, as we did for the alpha cobra toxin example, or we'll select on fractionated venom, as we did for the dendrotoxin example. In the case of the dendrotoxin, we may then have to do mass spectrometry as before to confirm or identify the target that we're working with. But ultimately, the idea is to funnel these through into target-specific assays, such as protease assays and so on, to identify our functional antibodies. Obviously, as you'll hear in the next talk, there's a desire to get cross-reactivity as well, to get more sort of bangs for your buck, and I'll leave that to Andreas to expand upon. This again is a trial that you heard about earlier in the day, in the beginning of the morning session, which is the one and only controlled clinical trial in Nigeria, looking at the effectiveness of this anti-venom from the Claudemiro Picada group and comparing it to another one. Again, in this paper, we've heard today different levels of lethality for envenomation. In this particular paper, they were using a number of 12% of the people who are bitten end up dying from this one. In the paper, they were measuring the coagulation and recovery from coagulation, as you heard earlier, and demonstrated that the majority of people who received the antibody restored their coagulation capability. And in fact, it also said in the paper that of the 200 people in each group, there were no examples of lethality. So definitely this antivenom does the job. However, it also has problem that we talked about earlier, coming back to the fact that you're injecting poor unfortunate with multi-grams of foreign protein. And I can barely read these numbers here, but it's better. So we're finding 25% of patients are showing some sort of early reaction, and with 10% of them being described as showing late serum sickness type reaction. So this still remains a problem with these antibodies, and some of the motivation for this project. And the last thing I would then say is that as well as using phage display and mammalian display selection techniques to distill out a set of antibodies that hopefully cover the majority of problems, another aspect is that the idea that we would then provide these back as chimeric antibodies. So, back in the 1970s, Cesar Milstein and George Kohler described the murine monoclonal antibodies, and these were the great hope for therapeutics in the developed world. But people quickly realized the problem of human anti-mouse responses to these, and then the next generation of antibodies out were chimerics, where you keep the variable domain, in this case of the mouse, and graft it onto a human backbone. And then, of course, we kept pushing to make them more and more human. So then we had in the late 1980s description of CDR grafting, so-called humanization. And then finally, in the 1990s, we had the generation of fully human antibodies from phage display and from transgenic mice. And as we kind of progressed from left to right here, we talked up the importance of being more and more human. But it's worth pointing out that the chimeric antibodies have been around for about 25 years, and they're still active and still making money, and people have many doses of these. And in fact, there are still chimeric antibodies being approved. The last one I'm aware of was 2 years ago. Europe approved another chimeric antibody. So my case would be that by the combination of using phage display to generate equine or llama variable domains, together with the presentation of those as chimeric antibodies, I think overcomes at least the discovery part of the problem. But again, I don't underestimate the whole scale of problem here. I've been able to move on from this modern recombinant antibody technology and actually turn it into materials that can be injected to patients. But each journey begins with a step, and I think in some ways the generation of binders is the least of the problems, and I think there are other areas that we're going to have to push and work on over the next decade to finally make a difference to the lives of people in the developing world. So I'd finish off again. I acknowledge people on the way through here, but again, thanks to Andreas, who brought the whole world of antivenoms to us. I'm grateful to my collaborators at Claudemiro Picada. Look forward to a fruitful few years working with them. I mentioned Anish, who was at IONTAS and is now CSO at Maxion. As I mentioned, he'll talk tomorrow. And also mentioned Jill Murphy's group at the Institute at the University of Cambridge, who provided us with great collaboration and multiple outputs. Thank you very much.
Thank you. Questions? Peter.
That was a beautiful talk. Really interesting. I wondered for the neurotoxin part, when we think about treatment and treating after, say, a snake bite, do you think the blood-brain barrier is relevant? Do you think getting things into the brain would help the efficacy, or is it all trapping it before it gets there? Or just thinking from a treatment perspective?
So, I mean, certainly there's the prospect of working peripherally, at least in the early stages of doing that. Andrea, I see you shaking your head. Do you want to add something?
One exception, toxins from snakes don't cross into the brain. There's one exception from Naja in the Caribbean. Maybe future exceptions, but only been found out of thousands.
Okay.
Thanks, John. Well, if anyone can pull this off, of course, it'll be you. But there'll still be the problems that were mentioned in the earlier talks about just the logistics of getting these things to people in the field. Are you planning to do something like have a really stable combination of antibodies that you could lyophilize, for example, and have them sort of specific for certain areas? So in certain areas there can be points with vials ready of these lyophilized products that can make them much more readily available.
I mean, this is, there are people who've spent a lot more time thinking and considering these questions than I have. Obviously, freeze drying is one solution. The whole cold chain is going to be a problem. That said, I've heard other people talk about the fact that with, solar panels and organization and so on, it may be possible to have solution antivenoms distributed more readily. We've also heard solutions about that are nothing to do with high technology, about having motorbikes at the ready and communication and people knowing where to go and so on. But yeah, you're obviously correct in that one. I think I also wonder if there might be, again, this talk is obviously all about antibodies. I think Nick was discussing small molecule solutions, some of the inhibitors of proteases from before that have maybe failed clinical trials for other reasons. So I think there's all sorts of possibilities and solutions, but arguably freeze drying might be one simple way to deal with it, but I think for me it would be better if freeze drying brings the potential for aggregation and other problems. Anyway, I'm rambling on a little bit there, so I don't really have the answer to that. I think, as I've said, there are multiple problems that have to be solved. That's just one of them. The other is just about the economics of it all and how to get there. But I think if you don't start the journey, you'll never get to the end.
Hi, John. I think it's working. Is that right? Yeah, I see you going into llamas. Do you see any benefit of keeping them as VHHs and then a cocktail of VHHs with IgGs? Maybe because of that smaller in-between size, would it help get...
So what you suggesting, Phil, are taking a VHH and turning it into...
And keeping it, so your cocktail will be made up of VHH plus IgG, and then you get a bit
yeah, of course, yeah. I think if one had a VHH, you'd probably just, keep it as an FC fusion, but there's no reason why you couldn't mix those with regular, 4-chain IgGs.
Just a short question, maybe I misunderstood, but there was example where you inject intraventricular in a brain. Why artificial situation if toxins are not going to brain?
I think it was a convenient way to demonstrate the effect of the dendrotoxin, which obviously has an activity in those voltage-gated potassium channels in the intracerebroventricular space. So it was I guess a sort of practical way of getting an experiment done to show an effect.
Okay, with that, I think we'll move on, and we have more time for discussion after this, so please hang around.